Is that all right? Okay. So hi, everyone. I'm going to try to be quick, so any soccer fans out here, though I don't suspect there's many, can go back to the World Cup. Okay, so I'm actually going to go a bit off topic, and actually let me introduce myself. I'm from the Victorian Life Sciences Computation Initiative in Melbourne, and I'm also a core scikit image dev. Okay, so I'm going to start by giving an update. If any of you were here in SciPy 2012, I presented a segmentation method for connectomics, so very large image volumes. And since I presented it, it was called Ray. I asked for a name. I wanted to rename it, and it's now called Gala. And there's this segmentation competition. That didn't work. Anyway, that's supposed to be rotating in 3D. And I submitted that as part of revisions to the paper, and it actually came in first, much to my surprise, really, since it was actually not even designed for that kind of data. So that's very small. Even I can't read it. But the point is we were first, and then this other team, 8 months later, came on top. And over the past few months, I've actually worked with a Princeton undergrad, Neil Donnelly, who's got us back to first place by adding some new 3D features and stuff. And the reason I'm mentioning this is because he is now touring South America and looking for freelance work that he can do remotely. So if any of you are interested, I had a fantastic experience working with Neil. And I've never met him, but just our communication was perfect, and we got a heck of a lot done in the space of a few months. So look for that. And the other thing that I wanted to mention is come to SciPy, come tell all your colleagues to come to SciPy. This is my second one. I also went to EuroSciPy last year. And when I came here 2 years ago, I had never done a GitHub pull request. And then I stayed for the sprints, so that's another thing, stay for the sprint. And these are my first 2 pull requests ever, and they're just like tiny little code fixes. And Tony and Stefan guided me through them, and as I said, so now I'm core dev in scikit image, so the community for scientific Python is so strong. We're always looking for new people, we're always helping people get up to speed, and yeah, so tell all your friends. Okay, so high content screening, what is it? Basically, it's reverse genetics on a massive scale. For if you haven't done genetics from sort of the historical perspective, forward genetics is when you have some phenotype, such as blue eyes, and then you do some kind of genomic mapping to try to figure out what gene controls that phenotype. Reverse genetics is the other way. You mutate a specific gene, and then you look at what the phenotype is. And high content screening is the extreme version of that, where you have a whole bunch of these 394 well plates, and on each of the wells you seed some cells, grow them for a bit, and then you add some RNA interference or a drug, a different one to each well. And so you're knocking down specific genes, and then you take pictures and look at how the cells respond to losing that gene. So that's the phenotype that you're measuring. And depending on what you label in the cells fluorescently, you will see different aspects of the phenotype. And high content screen in particular has had a very good track record over the past decade in drug discovery. And I was surprised to find that actually more drugs were discovered in the past decade through high content screening than through target-based approaches, so protein modeling, molecular docking, and so on, even though vastly more resources were devoted to the latter. So that's really impressive. So why do I want to do unsupervised clustering of these images? So the main reason is cost. It's about $250,000 to run a screen. And typically the output is a single gene list. So let's just say I'm looking for genes involved in cell proliferation, and in all my wells, I just count the number of cells that are present with or without the treatment. Then I rank the cells according to that measure. So this is from an actual paper. and we just look at the ones that positively regulate cell proliferation or negatively regulate it, and get some hits. But what if you have a well that looks like this? That's something really interesting going on, obviously, but if you're only looking at the cell count, you're not going to pick it out, and that could be a Nature paper hidden in there. Never mind that plot is from a Nature paper. Okay, so the key thing about clustering for high content screening is extracting features. So if you're not familiar with clustering, this is what you want your data to look like. A 2D matrix of features and samples, and then you cluster your samples according to how similar their feature vectors are. And so what you want is to take your images and produce, take measurements from that image, and they can be all sorts of things. So this is from a science paper, 2007, and I don't really know what they're measuring in that, but essentially just any kind of number you can pick out from a cell, you try to get it. Our particular screen was actually extremely difficult. So these are muscle precursor cells, and we are interested in cell fusion, so you end up with big, long, multinucleated cells. I think they're called myofibrils. And they're all intertwined, as you can see, and there's just no way I'm going to try to segment those cells so we can produce accurate features, such as what the biologist would like, so number of nuclei per cell, etc. If you can't even find the cells, you can't produce those numbers. However, there's this quote, which might be apocryphal, but I really like, so I'm going to give it anyway. So from Fred Jelinek: "Every time I fire a linguist, the performance of our speech recognition system goes up." And the takeaway from that quote is you can actually do quite well with random features, features that are uninformed from an expert perspective. And I'll mention that this is Christophe Marcel. He's the one who generated the screen. So don't care feature extraction. This is my approach. And so we have one channel there. And all I'm doing is doing a threshold Otsu, which gives me some level, and then looking at connected components of pixels above that threshold. And so these aren't cells. These are sometimes groups of cells. These are sometimes noise in the image. And some images are a bit more challenging. They're more light and dark in the image, so I also do an adaptive threshold. And as you can see, the two pictures of the thresholding for one image look quite different. But again, I'm not going to care. I'm just going to extract features as if all of those things on the top were cells and all of those things on the two bottom things are cells. So what do we actually measure? We take an image. Image has 3 channels. For each of those channels, we have 2 different thresholding methods. And for each of those thresholding methods, we use scikit-image's region props and measure 8 different properties. So things like eccentricity, solidity, which is how much, if you draw a box around your shape, how much of that box is filled with pixels of the shape. So a square will be everything, whereas anything that's very spindly will be very small solidity. And I also count the number of objects. So how many objects are there in the image? And so each of these things is, except for the nom-objects, is a per object measurement. And we have lots of objects, but ultimately we want to reduce the image itself to a vector, not an object. And so I take 5 quantiles of those quantities. So if we have a 100 cell, we take the cell with the 5th largest value for that property, 25th, 50th, which is the median, etc. And in addition to that, as a concession to Christophe, I tried to do some smart things about things that a biologist would care about. So number of nuclei per cell. Again, here cells are not a real entity. They're just these thresholded objects. The distance to the nearest nucleus, because once the cells fuse, you can either have the nuclei evenly distributed or they can be clustered together, and that's important to biologists and so on. So the feature extraction I can't really demo. I did it on the cluster. And so Mary is the name of our cluster. And all of our little subclusters are named after Australian rivers. So I have all the images there. I grouped them by plate. So you saw there's this 384 well plate. There's tons of those. And then each of these is a job. And I compute the feature maps for all those images. And I get a pandas HDF5 file of the features. And the file is keyed by the file name of the image that produced the features. And that's important later on when we want to look at the images. And so then I fetch those to my local machine and concatenate them and do some clustering, which we will do a demo of. So let me mirror the displays. So I have this notebook both unfilled and filled, and if we have to, I'll go back to the, okay. And I'm also using Damian Avila's live reveal, which is really cool, I think, for showing code, as you'll see. So this is a notebook running in slideshow mode. I'm going to make that bigger. Sorry, the font's a bit small. Okay, so the first thing to know is that I keep all of these mappings in a MongoDB database. Basically, we want to keep track of a gene. How does that relate to an image file? How does that relate to a plate well coordinate of the sample? And I used to do this on a Python dictionary, a whole bunch of Python dictionaries going each direction of these mappings, and it was a huge mess. So MongoDB, if you don't know about it, is this big distributed database. It's open source, huge support, Python bindings, and it's, in particular, for someone who's very naive about databases, such as myself, it's an extremely simple model. So a document or an entry into this database is just a collection of key values, pairs, and you can search, give me all the documents that for this key have this value, and so on. So it's really nice. So first thing I'm going to do is start the MongoDB daemon. Yep, so that's running. So there's a thing in the background now that's got access to my database, which is on disk, and will respond to my requests. And then I will show you what a MongoDB looks like. So this says from PyMongo, which is the Python bindings for Mongo, import the Mongo client, and then a collection is just two nesting levels down of database, and in each database you can have multiple collections. So these are just all my wells. Okay, and then let's just say I want to find this particular gene. I just say collection.find1, and then I pass it a dictionary, gene name, this. And you can actually put as many keys into this dictionary as you want to match more and more precisely what it is that you want. And this, by the way, will always return the first hit, even if there's multiple hits. So that's a nice way to go about it. Okay, and so here's an entry into my database. These are the keys. The ID is the plate number and the well coordinate. And I've got the gene accession number for, I think, UniProt, gene name, etc. So that's everything we want to know. And also the location of the file on the cluster. Okay, so then we just do some pandas. As I said, I've already downloaded the feature maps from Mary. And so here I'm just taking all of the feature map files, reading HDF from pandas, and concatenate them into a big data set. And that's what it looks like. By the way, I missed how to do pretty HTML printing of pandas, so that'd be something neat to know for later. Okay, so then we do the scikit-learn stuff. This is really simple stuff. Taking a standard scalar, fit transforming the data. That's my x. I'm deciding I'm going to use k-means, so I need to know how many clusters I want, and I'm just going to decide that I want an average cluster of size 10, and so the number of clusters is the number of samples divided by the average cluster size. That should be quick, yep. By the way, feel free to stop me if you have any questions about what I'm doing. Okay, this is a bit of an aside, but the only thing, as I mentioned, everything was keyed by file name, but there's a catch, which is that the file name is where the feature mapping was run, which is on the scratch directory, which is not where the files actually end up in the end. So I have this function, which is part of my screens submodule, and this is where every time if you have a screen, you're going to put anything that's screen specific. Any code that's screen specific, you're going to put it in this package. And so then I'm just going to get this function that transforms from file name to everything that the file name gives you, which includes the plate and well coordinates. And so if we go down, oh, I'm in edit mode, I can now replace the index by the actual coordinates of the image. Oh, that should be rendered. No, I'm stuck. Okay. So, this is just getting me from the Mongo database. If I give it my plate and well coordinates, give me the documents from that query. And what am I doing here? I'm just looking at the cluster sizes because for the demo, I don't want a giant cluster. I'm going to show you tiny clusters of a few images. And as you can see, k-means produces quite variable cluster size. So I'm just going to find things that are 6 and 9. And by the way, these are all premeditated to show you the prettiest pictures I have, but I'll assure you that the conclusions are unchanged. Okay, so let's actually have a look at some of the clusters. So here I'm just getting the genes for this one particular cluster, 136. And these are all the gene names. So one particular thing which is nice that you'll notice is that the data actually has genes in duplicate and triplicate. And here you see that we've recovered 3 gene pairs. So just by clustering the images, we knew nothing about what the images were. We've found images that corresponded to gene duplicates. So that's pretty nice. And then I'm using Paramiko, which is an SSH library, if you haven't heard of it. And it's way, way faster than, say, using scopy as a subprocess or whatever. And you can basically do anything that you can do over SSH using this Python library. So I highly recommend it. And you can have a notebook where you're using your agent and you don't have to type out your SSH password in front of an audience of forever, since these videos are being recorded. Okay, so I am now connected to the VLSCI server. I'm not actually going to rely on the network. You'll notice this little, if the file has already been downloaded, just don't try to re-download it. So all the images are already on my computer. However, this particular function will get me images if I say, give me all the images corresponding to this particular gene. So I'm getting the images for that particular cluster, along with titles. And let's have a look at them. So this is just a function that shows many images together. I wish that were more visible. Can I do any kind of zooming? Yes, I can. Awesome. Okay. So you don't really have a background for how similar these images actually are, but at least you can see that the clustering has retrieved the same gene twice, multiple times. So that's pretty cool. And if we look at another cluster, you will see just how variable these images are. Okay, so let me go back and forth. So that's those. And these ones. Okay, so these are way more spindly and so on than the images that you saw earlier. So I think that's pretty cool because, as you saw, I was doing kind of the dumbest thing possible, and yet it still worked. So I'm going back to the presentation, which is here. Okay, so what do I actually want to do with these clusters? So I should have mentioned at the start that this is all very preliminary. So I'm not actually looking to make it easy for biologists to run these analyses. So there's actually a software called Cell Profiler that some of you may be familiar with that does that. But the problem with Cell Profiler is that it doesn't allow you to express, if you know what you're doing, it allows you to express everything that people have programmed into it, but not anything outside of that. And so my goal with this is to make it easy for bioinformaticians to run similar analysis and do some cool post-analysis stuff. So in particular, the things that I want to do with it, first is improve the image preprocessing. As I said, I've cherry-picked the images that look best. Some of them just are chock full of artifacts, and we need to deal with those. When you're going to be running 45,000 images, or they're going to 500,000 and several million now, you're going to get some images with artifacts, and you need to be able to deal with those intelligently. Clustering, I have like zero experience with it, and I just did k-means and it worked. But I'm hoping that someone here has more experience and will tell me some nice things to do with it. We also want to remove any artifacts. So for example, some entire plates have imaging artifacts and so on. So it'd be good to take that into account and normalize each plate separately, which I'm not currently doing. Finally, do a whole bunch of downstream bioinformatics, such as once you have the cluster, do some gene function prediction. If you know the function of all the genes in that cluster and you know that this new gene is having a very similar effect on the cells, they all look the same as the other ones, then you might be able to transfer that functional annotation. And also, I would really like for this tool to be used for interactive data exploration. So maybe Bokeh would be a really good thing for this. Again, I don't have any experience with this sort of stuff. But for example, it'd be cool to select a gene, have a researcher, he knows what gene he's interested in, and then look at all the nearest neighbors in this feature space to figure out genes that have similar effects. Additionally, you want to be able to plot features and look at extreme values of those feature distributions and see whether a particular feature is measuring something that you're interested in. Because, as I mentioned, the features are very sort of raw and not trying to get at anything in particular. Okay, and that's everything. I also would like to announce that there will be an unofficial scikit image sprint on Friday, and by unofficial I mean I really hate locking myself in a basement in beautiful Austin, so it will be above ground somewhere, and preferably outside, but I'm sensitive to people who don't like the heat as much as I do. And tutorial solutions, if you were at the scikit image tutorial, will be coming soon. Tony's put his up. Mine will be in later today. Just keep an eye out on the repository, and you can also follow the skimage hashtag on Twitter. Thank you.